All right. Hey, everyone. My name is Max. Oops, I have the stream playing at the same time. It's distracting. Okay. Sorry. Hey, I'm Max. I have been working with OpenInsulin for almost a year now on the science and the wet lab side. Yeah, I am, or besides that in my life, I'm a PhD student at UC Berkeley studying cell biology.
Okay. Hi, everyone. My name is Yann. I'm a French dude who went to the US to follow my wife. She was doing a postdoc, and then I found this project, and I started being involved in the lab. So I've been in the project, I think, 3 years now, and now I work mostly on developing the protocol to produce insulin. So hopefully David will be able to join soon, but I guess I will try to find a way to invite him again, but maybe you can start presenting the project if you will.
Okay, so yeah, as you may have seen from what we're advertising for today, we're having a science-focused community discussion. So, all of us talking today have been involved in more of the day-to-day lab work for OpenInsulin. So we're gonna chat about that today and answer questions people have about the science. But just to start with a general overview, if this project is new to you, so the OpenInsulin project aims to develop an open source protocol for producing insulin in community laboratories. So this started out of our home base kind of counterculture labs in Oakland, California. And since we've begun, we've had the pleasure of bringing in some more collaborators, including, Yann used to do the lab work in Oakland, but has since moved to Baltimore and is doing some work when the lab's open at a community laboratory there called Bugs. So the actual work, there's many different directions of work to be done for the project, not limited to legal work, communication, and so on. And so, there's areas of the project people are working on that, but we're involved in the lab work. So the types of work that we're doing on a day-to-day basis involves some cell culture, since we're trying to produce, engineer different species of microbes like yeast and bacteria to produce the insulin. And then there's protein purification strategies of, once we've grown up our cell cultures, we need to extract the insulin that's being produced by them. And there's, following that, there's methods of validation involved, like HPLC and mass spectrometry. And then finally, formulation into an injectable medicine. So I'd say those are the main kind of branches of what's going on in the lab. So we can talk a bit more about each one. I'm not sure if you had anything to add to that, Yann, on a more general level.
No, I think it's good. I think it's the most the big class of thing that we have to do. Yeah, I think so far we are more we are starting to be at the phase where we need to focus on the quality control kind of aspect of it. But yeah, I think it's a good summary of what we are doing. I don't think there is a question yet. Maybe we can explain a bit more, I guess. So far, for the main project, we are working on 2 different types of insulin. So we are working on Glargine, which is a fast-acting. No, Glargine
Glargine
is
is the
not.
slow acting.
Yeah, slow acting. Damn, I'm always mistaken with two. It's a slow acting version of insulin, and we also are working on Lispro, which is the fast acting one. So we try to produce and purify the Glargine from a yeast host, and the Lispro from we try to express it on E. coli, basically.
Yeah, and we're we're not starting from scratch here, because I mean one of the reasons why like of all things to apply like community biology to, and looking for alternatives from the like concentrated pharmaceutical industry model is that like the thing about insulin is it's been isolated over 100 years ago, and the techniques to produce it are well worked out by now, and there's decades and decades of research behind it. So all we're trying to do is adapt some of these well established methods to being produced in a community lab, and show that it's absolutely ridiculous that something so well understood is still costing so much to patients.
Yeah.
So with we're starting with these 2 variants of insulin, Glargine and Lispro, because there are some production protocols already published in the scientific literature that are not under patent protection. So that's I just want to emphasize that we are doing the science to figure out how to make these things in the community lab, but like these things are already well understood, and it's there's a high likelihood that we can get it to work in our labs.
Yeah, and I think it's also important to mention that we don't aim that the community lab itself will be the place where we produce the insulin, but we are mostly developing the protocol that can be used afterwards to produce the insulin. So it's something we have often, that people think, oh, we want to develop a protocol that people can do in their garage, and it's not really the way we see what we are doing. I think the emphasis is more, oh, we are doing development, and when we'll have a robust protocol, then it can be used to produce insulin in manufacturing facilities, basically. Mm-hmm. So we have a few questions. So why do you produce one form in E. coli and the other in yeast? Which really technical.
I mean, my answer to that is we're going off of what has been published already and what we know has worked. Lispro has been produced in E. coli, and Glargine has been produced from yeast. So there's probably other reasons included there, and different like advantages and disadvantages to each.
Yeah, this paper when they express Glargine in E. coli, but then it goes into the inclusion body and you have to do refolding process, which is not really a manufacturing friendly process. It's quite difficult to do that, especially if we want to focus in small and medium scale. Then refolding is not really easy to do in that kind of scale. But yeah, but at the end we kind of try to reproduce papers that we find. So if it's working as described at the paper, we try to not change too much from the paper.
Maybe we can go into a bit more detail about the issue with inclusion bodies and refolding. Right, so insulin is a protein, and every protein, in order to be functional, has to adopt a specific fold, or like, that's another way of calling like the structure. Like it has to have a certain three-dimensional structure that it starts as a single polypeptide chain and it has to fold in a certain way in order to be functional. And that's true for insulin. So the structure of insulin, it has some features that are really important to consider, that they're folding correctly. It has these types of linkages called disulfide bonds, which is covalent bonds between two sulfurs on different amino acids along the polypeptide chain. And there's three total, right? there's one intra chain and two interchain ones. So each of those, like that, as insulin is being made in cells, the chains are put together first, and then the disulfide bonds are linked together. So you're the protein biochemist. You know better than I do.
Normally they should kind of in the way they are produced, the disulfide bridge start forming, which is kind of the complicated part. Is if it doesn't happen that way, then you can have a mismatch, and then you can have not good form. But the dynamic of those bond forming is really important for the 3D structure of the protein, which agree.
Right. So that if we're engineering cells that don't normally produce insulin to produce the insulin, it's likely that unless we're careful about how we're designing that strain, then it's easy for it to get folded in the wrong way and then not be immediately functional. So like we can either design the engineering strategy such that it most of the insulin is folded in the correct way as it's made, or we can worry about that folding after we harvest that protein, which is what the refolding
is. And the issue of this process is the yield of so basically the idea, you take the protein which are not folded the proper way, and then you how you say that, you unfold it completely, and then you put it in a condition when it will refold, and you hope that it will refold properly. And the percentage of protein which will refold properly are quite low. And also it involves dilution, usually, I think it 1 to 1,000, 1 part for 1,000. So if you have, 1 liter concentrated of your protein, you need 1,000 liter to do the dilution to help for the folding. So you need big volume kind of system, which is why it's not really suitable for small scale.
So we have a question about that. How do you test for disulfide scrambling? So I guess, how do you know that the correct disulfide bonds are there in the structure?
Yeah, it's good question. I think the easiest way is still mass spectrometry, because the difference of mass between a disulfide bond and the two sulfur function, SH, the difference of mass will be enough to see if you have a disulfide bridge or not. You also can do reverse phase chromatography, because the 3D conformation of the protein will be different if there is the proper bonding or not, and you should see the protein behaving differently during the chromatography, basically.
So just to explain a bit more about those techniques, so mass spectrometry, this is a technique to determine what the actual like chemical subunits are in a bigger macromolecule. It's, I don't know, maybe this is an abuse of terms, but I think of it kind of analogous to, like, for if you want to know the sequence of DNA, you can do DNA sequencing. And then if you want to know the sequence of protein, sometimes mass spectrometry can kind of give you that analogous result. And the way it works is,
yeah.
Yeah.
Close enough.
It's not exactly the same,
Yeah.
but it can give you that information. The way it works is you have you give the machine your sample of protein, and then I guess there's a few different types of mass spectrometry, but the one I'm familiar with is it shoots some high-powered radiation at your sample, which blasts all the individual or different fragments of your protein sample into a detector, and each individual, whether it's like an amino acid or a smaller group, will have a specific distance that it travels through an electric field. And so by measuring that distance, you can understand what that chemical group was. So that's one way to actually get an idea of what is in your sample. And then so there will be differences based on which amino acids have disulfide bonds or not.
Yeah, the accuracy of this kind of technique in the atomic mass range. So it's even atomic mass, one atomic mass, it's already for the low end mass spectrometry. So usually it's even multiple decimal below that. So yeah, it's really accurate. So you can even the difference of 2 hydrogen, you will see it in the mass of the molecule. So you will not have an information of if there is mismatch or not, but you will have information if you have the disulfide bridge or not. Mm-hmm. Then for the mismatch, I would say the chromatography, in which kind of depend of the shape of the protein, will give more information, because if there is mismatch, the 3D confirmation will be completely different, and you will see the difference on a chromatography reverse phase.
Can you talk a bit more about what like a chromatography run or experiment actually looks like? Like what?
Yeah, so
what does the machine do?
Yeah, it's a really generic term, which basically kind of describe all technique which used to separate molecules. And in that case, the reverse phase chromatography is, if I'm not mistaken, it's not really my expertise, but it's by hydrophobic interaction. So you put your protein in a solvent, and then you run it in a column which will bind. If the more the hydrophobic your protein, it will bind, something like that. I don't remember the which direction it is. But the idea is the hydrophobicity of the protein depends on its conformation, because it will depend on the exposure of the hydrophobic core. I don't know. I don't want to be too technical, but basically protein usually have the outer layer, which is charged or hydrophilic, and the inner core of the protein is hydrophobic. So according to how much the inner core is kind of exposed to the outside, then you will have different binding to this. So the way the shape, basically, if I want to make it simple, the shape of the protein will induce different chemistry properties, and then they will behave differently with chromatography technique. So the chromatography could be hydrophobic interaction, but it also can be charge interaction. It can be with specific affinity. Like, there's a lot of different chromatography, but the behavior of the protein is related to its shape usually.
Yeah. So you have a column that you feed your solution of protein through, and the specific type of column depends on what physical or chemical property you're trying to separate out things by.
Yes.
But yeah, once you know which way you want to separate things, then you feed your sample through there, and then every different type of protein will have a different profile of which, like, how much they get stuck to that column versus how much they are able to flow through.
Yeah.
So that's another way to detect pretty minute differences in protein structure based on how they interact with the column. And so
there is another way that we can do that, is doing activity tests and look at the total amount of insulin we use and look at the response, and then we can estimate how much of the protein were properly folded, because only the properly folded insulin will trigger a response from the cells. So basically, the activity test, it's you grow some cells which are designed to have a response when you put insulin, so they have insulin receptor and so on. And you put insulin, and then you calculate the glucose intake. So with this kind of assay, you can know since you know how much insulin you will use, then you can estimate how much properly folded insulin you have at the end.
Yeah, and that gives you like a unit that's actually used for like medicine, right? Like, you usually see the concentrations in terms of like units per mil or something.
Yeah.
And that's a general term for functional proteins and enzymes and stuff, that there's usually they come up with some definition of what 1 unit means, of like how much activity there is. And then once we have that measurement, then like people usually don't care about how many molecules are actually in your solution. It's just like the concentration of activity per volume is.
Yeah, unit it's a weird unit system, because it's not completely consistent from all the enzymes. So at the end, you have to focus on what is a unit for insulin and what it really mean. But it's
Cool.
How do you know that you are actually producing insulin? I mean, this one, it would be, the mass spec, or the glucose intake assay. Or we can, and this is something we were planning to do before the coronavirus pandemic, is to do Western blot, which basically you can buy antibodies which bind to the insulin, and antibodies bind to specific shape. Actually, it's more shape related than sequence related. So you bind you buy antibodies that can react to insulin, and then you have another solution that can allow you to see if you have a signal or not. So if you have really the insulin, then you will have this binding, and then you will reveal it. So this is something that we were planning to do.
Also a very standard lab technique that doesn't require much fancy equipment.
Yes, yes, it's pretty standard. We have another question. Do you have any FDA hurdles before this is available to the public?
Yes.
Short answer, yes. Yes, it's. I mean, the science team don't focus too much on that. We really focus on producing insulin. But yes, there is a lot of FDA issue, and there is a lot of effort from other member of the project to find what would be the best way to go through these FDA situations and at the end manage to produce and distribute it to the public. Yeah, I'm not sure I'm really the most qualified to talk about this, but I don't know if you want to.
Perhaps we could do another discussion on a different weekend, more specifically about the regulatory hurdles, because I agree that's also not my expertise, and I'm afraid to say anything that's totally wrong.
But
there are people in the project who are focused on that.
That we know it's a big issue, maybe the biggest issue of the project already, and people are trying to address it. But yeah,
like the lab stuff, we know it's possible.
Yeah, exactly. The protocol, we can do something. But it's political. It's different ballpark completely. Yeah.
But I mean, just to expound a bit more on that subject, I mean that's one of my personal main reasons why I'm so invested in keeping this project going, because even if that is the final roadblock of producing or making insulin available to the public, we're still able to make a massive political argument of look at us in this community lab, we've made this medicine that pharmaceutical companies tell you they need to charge so much because of all the R&D that goes into it. And so it's no, that's not the problem. That's not why it's so expensive.
I mean, there's good argument with there's a lot of stuff happening right now because of coronavirus, and all these big pharma are trying to do the program to suddenly they can lower the price if you justify that you have specific condition. It's always been tested, but there is a lot of this kind of program which is kind of a way to admit that there is no reason to charge also that much, that they can do it, they just don't want to do it. So
absolutely.
Another point also is FDA is important, America is important, but it's not only about the insulin issue in America, but we try to think about the project as a global solution, and we want also to make it available in place where there is a lot of shortage and issue, which is not necessarily the price, but sometimes it's just nobody is really locally producing the insulin. So then there would be less regulatory hurdle, and we have more chance that it can happen in those places. So FDA is a big issue though. But
some more questions.
As you move from research to development and eventually to a commercial product, what quality control assays are you going to rely on for releasing a batch? Well, it's a good question. I think we can say we're still in the research phase, and I don't know what would be exactly the difference between research and development, but still, because we didn't really confirm we have insulin, I think we can say we are in development right now. For the quality control, sorry, do you want to expand on this?
No. I think you know more about quality control.
Yeah. For the quality control, It's a really complicated one. I think the way we see it right now is trying to find third parties who can do that for us, and then we don't have to do it in-house. So maybe find a way to find a place where we can send a batch, we produce, and then they run tests on it, and then they say, oh, it's approved or it's not approved. Because the issue is the issue with quality control is it's kind of you need really mass spec or expensive apparatus that in a community lab it will be difficult to make it running, and sometimes for safety reason, because some of these apparatus need a huge tank of liquid nitrogen and stuff like that, and that we don't have the resources to do that, and the resource to make it safe to use this kind of equipment. And sometimes just money wise also, those equipment are really expensive. So I think it makes more sense to just focus on the production of insulin and then find a way to outsource the quality control.
Cool. And yeah, speaking of the research and development, that reminded me, we haven't really talked about where we're currently at.
Oh yeah.
In the research. So I think people are probably curious about that. And yeah, since it's been a few weeks since we've been in the lab, I have to rack my brain a bit about it. But as we mentioned at the beginning, there's two different forms of insulin. We're working on producing Lispro, which is a fast-acting variant, and Glargine, which is a slower-acting variant. And so I guess we can start with Glargine. We're going off of a published protocol that produced Glargine from the yeast species Pichia pastoris. This is a very commonly used microbe for industrial purposes because it can tolerate producing a ton of protein, which is what we want. Since we want to maximize yield for how much the volume of microbes that we're growing. And the general strategy there is to genetically engineer this yeast species to produce the pro-insulin form of Glargine. So one detail that we haven't talked about yet is the fact that like insulin is always first synthesized as pro-insulin, and then there's some post-processing steps that happen in the cell that that cleave some parts of it to turn it into the active insulin, which gets secreted. So we need to do a similar thing for our engineered Glargine and Lispro, but so just still focusing on Glargine. So we tell the yeast cells to synthesize the proinsulin form of Glargine and secrete it into the liquid medium that it's growing in. And the fact that it's being secreted makes the later step of purification much easier than it would have been if it weren't secreted, since if we just like, once the cells are growing, we spin them in a centrifuge to have all of the cells in a pellet at the bottom, and then the supernatant is the extracellular growth medium that contains insulin. So if we can just purify the protein from that supernatant, there's a lot less other protein to separate it out from. So it's generally easier than having to crack the cells open. And yeah, so we pretty much said what comes next is we separate out the cells from the medium, just take the medium and put it through a machine called an FPLC, which is fast protein liquid chromatography.
Yes, I think it's
There's so many acronyms that are like very similar but mean different things.
I think there's two different, but it doesn't really matter.
Yeah.
Protein purification system.
Yeah. Which the way that works is it starts with our supernatant solution that contains everything that was in the growth medium, and we feed that through a column that let's see, we use an ion exchange column first, right? Yeah. So the way that works is we're relying on like knowing what we expect the surface charge of the proinsulin glargine to be. So we set up this column that should specifically bind to I mean, I shouldn't say specifically, but it's biased to bind the proinsulin glargine and let other things flow through. And then we're currently just testing it out with that one step of chromatography, but in the future we'll probably implement more purification steps depending on how that looks. And then we we see what comes out of that purification and then test whether it's the real deal. So where we're currently at with that is so I guess there's two main like technical hurdles we're dealing with right now. So the first one is optimizing our like genetic engineering, since whereas with other microbes, it's people generally like have their gene of interest on a plasmid, which is like its own little piece of DNA that you can easily add to your cells. For this species Pichia pastoris, instead we need to do a genomic integration, which so instead of having a separate piece of DNA containing the Glargine gene, that has to go into the cell's actual chromosomes. And that's a little more random process that requires more like doing that to a bunch of different cells and then screening individual colonies to see which ones happened to integrate that gene in a good spot to produce a lot of protein. So that's one area we're actively working on, is doing that transformation and screening. And so the term transformation just refers to inserting our gene of interest. That's one step. And then the other side of things is looking at the protein side. So one added layer of complexity here of like how we're working through things is that while our end goal here is to have the Pichia pastoris yeast engineered and producing Glargine, we can still produce some Glargine using a different species that's easier to engineer. So that's what we've been doing. We've been using this other species of yeast called Saccharomyces Cerevisiae, which is, you might know it better as baker's yeast. And so this species is a lot easier to engineer, and because it does hold on to plasmids really well. So like in parallel to doing this Pichia pastoris engineering, we're also working with the Saccharomyces, which is already producing some protein. And so we've put it through that purification process and recovered some protein that looks like it should be Glargine. But the next step is validating that, which has been pretty technically difficult so far. So yeah, we're exploring different ways of doing that validation. So we're looking around for facilities that can do mass spectrometry to send samples to. We're also building or getting a HPLC machine to work at our counterculture labs headquarters. And we're also, like, in the meantime, those are like pretty technically challenging things. So yeah, as Yann mentioned, right before we were no longer able to enter the lab anymore, we're going to try some Western blots, which is a lower tech way to detect that the real protein is there.
Yeah, basically for both we have really kind of low tech standard experiment results, which was really promising. That it showed that we might have the proper protein for both LISPO and Glargine, and we kind of struggle to have access to some of the higher, more expensive type of equipment to confirm that we have the both protein. There's two questions. I will start with one, FDA related. What are the fees for an FDA approval protocol? Are you planning on setting up a fund for that matter? And what is your expected timescale for commercial release? Yeah, it's a bit out of the expertise again. I think, yeah, we will do fundraising specifically for FDA approval protocol. I guess it's in the million kind of range.
Yeah, I think I remember Anthony mentioning like the biosimilars path is like a few $1 million, and that's the cheapest option.
Yeah, it also depends on the classification, and the classification of the drug can, and there is a lot of talk that they want to change it, so it can reduce the cost a lot. But yeah, it would be, I guess, in the range of million. The expected timescale for commercial release, it's a complicated question. I mean, I would say, I don't know, years, but how many years, I have no idea. It depends on a lot of things, and a lot of things are not about technical issue, but more political issue. So there's a lot of story of people trying to create generic insulin in the US, and at the last minute, they didn't have the FDA approval without any explanation. So we don't know if we go through this kind of process. As our guess, it's if we have enough popular support, then it will help us to go through the whole process. But it's nothing is the given for this kind of question. Yeah, do you want to add something?
I mean, I want to emphasize, yeah, it's going to be mostly a political issue, and the more, I don't know, angry people we have supporting us, I think the more likely it'll be to move forward more quickly.
Yeah, I agree.
But that's pretty, I don't know, I can't predict how long that'll take.
Yeah, I mean, part of our thinking is that when we will confirm that we will have insulin, and when we will confirm it, then it will maybe help us to have some more political momentum to push through. But there is a lot of uncertainty in the equation, so it's difficult to tell. Is there a reason you don't use E. coli 20 to produce recombinant human insulin? E. coli 20. I don't know if it's. Do you know E. coli 20?
I haven't heard of that. I'm assuming it's a strain.
Yeah.
I haven't heard of that specific one.
Yeah, me neither.
So we are producing a different type of insulin from E. coli. So the E. coli Lispro, which is, or sorry, the insulin Lispro, which is fast-acting insulin. And I can talk more about our strategy there. So Jan had mentioned earlier, for a lot of industrial scale production of insulin is in E. coli, and they are when the protein is synthesized, it like E. coli naturally doesn't like having a ton of extra protein that it doesn't have any use for, so it sticks them into these compartments called inclusion bodies inside the cell, and that produces an issue of extracting the insulin back out, since when they're in inclusion bodies, they're probably not folded in the correct structure, and they can be not very soluble too, which you need something to be soluble in order to work with it. So we're taking a different strategy for producing from E. coli, which is directing the insulin to be secreted into the periplasmic space. So a little background about E. coli is that these bacteria have two different outer plasma membranes. So they have, it's called an inner membrane and an outer membrane. And in between those two membranes is called the periplasmic space, or the periplasm. And what's nice about that periplasm is the chemical environment is different from what's in the rest of the cell, which means that it's, I always get oxidation and reduction confused. It's an oxidizing environment.
Yes. Yeah. It's making the sulfide bridge. Let's say it that way.
Yes, it's an environment that encourages disulfide bonds, which is what we need for the correct structure of insulin. So that way we can have most of our insulin already correctly folded before we extract it. So we don't have to deal with like the refolding problem. But that also brings the added complication of we have to be really careful when we extract from the cells, since like they're in this delicate chemical environment in the periplasm, and that means when we are finished growing the cells and we want to extract the insulin, we have to be careful to only break open that outer membrane to release the periplasm, but leave the inner membrane intact. Since if those two compartments mixed up as we're harvesting them, that could increase the chances that the insulin that was there is gonna unfold and become insoluble. So that's kind of a nice transition to where we are in the on that side of the project. So we're going off of another published protocol for producing Lispro in E. coli, and so we did that genetic engineering, and we know we're producing protein from E. coli, and we're currently optimizing the strategy to harvest those cells and just break open the outer membrane. So I believe where we left off was we did kind of some very crude harvesting, and we've been able to.
Max, just to finish on the question, because I find a paper where they talk about the E. coli 20. So I will just briefly say something about the issue. I put the paper on the Zoom if you want to look at it. The issue is the strategy they use. So maybe there is other paper, but with the one I found, is that it's a collision body type of strategy. So we have to do refolding if we follow this strategy, and refolding, it's not really small scale friendly. The second issue, and this is something that could be investigated, but it seems that they patent it. So we try to find strategy, of course, which are not patented. So you will find a lot of paper when they say, oh, we find a really great new strategy to produce insulin. But usually then you check the author and you look at the patent, Google patent, and you will find a patent linked to a startup or linked to a big pharma. And then it's a liability for a project, because we will be linked to their approval for us to work with their material. So even if it's a license, you never know what can happen. The big pharma can buy the company and then say, oh no, you can't work with it anymore. So we really try the most we can find strategy which are not under any patent. So this is also why we follow this strategy for the E. coli, because there was a patent, but it was seized. It was abandoned. I don't remember the terminology, but it was in the public domain, basically.
And yeah, that reminds me that if you're interested in volunteering for the project remotely, I think that's a task that is in high demand for us, is kind of combing literature and checking for, like, we get ideas all the time of, different ways we can approach the engineering and purification and all that. And, every time a new idea comes up, where it's like, wait, okay, are we going to get sued for that?
Yeah, exactly.
And yeah, so we can always use help with kind of that type of literature digging and seeing
I mean,
which ideas are viable for us.
The paper seems interesting though, but yeah, I'm not sure we can use it. It seems to have really high yield though, but yeah. Yeah, they should have worked more on the figure though, but I'm bitching a bit. Yeah. Yeah, anyway, so yeah, this is kind of and you will find a lot of really interesting, innovative way to do insulin, but there's a really practical concern, which is, can we really use it in the context of our project? But yeah. Sorry, I interrupted you.
Oh, I forgot what I was talking about. It's fine. I was just looking at other questions we have.
Do we have more questions?
There's one we skipped over. What's the difference for science in the community lab versus academia? So I guess I'm currently doing both. Well, I don't know. I guess the first difference coming to mind is what we just talked about. I mean, I don't know if it's necessarily about being a community lab, but just for academic research, like, I am not worrying about pissing off like pharmaceutical companies. I'm not trying to make a product. So yeah, I feel like I can focus more on the actual science and not be limited. Whereas for this project, that is a huge consideration, and we don't want to waste time trying to develop strategies that will be prevented from using. I mean, there's still a value of trying things out and learning from them, even if we can't actually use them at scale.
The dynamic, it's really different also, because the idea is also to try to spread scientific knowledge and to teach anybody, not really taking into account the scientific level of anybody, but everybody is welcome to come and try to learn. So the logic it's a big difference. Oh, so Dave responded to the question. We had the question, what's the yield for the 2 strains you are currently working with, and what's your practical goal for yield? I think we are in the range of milligram for Glargine per milligram per 50 ml, something like that in this range, which is I think we kind of the best would be to aim for gram per liter basically, but this is something you can reach in a bioreactor, in fermenter condition, and in flask, because you will not have the same density of cells, that it would be way more difficult. So yeah.
Right. Yeah, just to reemphasize that, in the research we're currently doing, we're growing cells to optimize like speed and ease of doing the experiments over yield. So we're growing them in just flasks, shaking in an incubator, which is pretty easy to grow cultures in, but the really like yield optimizing way of growing microbes is in a bioreactor, which can it's like this. I've actually never seen one working, so I might be describing it incorrectly, but it's kind of a self-contained environment where there's like a stirrer in it and like some feeding tubes for like keeping all of the nutrients at a constant high level. So like the density of cells is, I don't know, it can be around like 30 times higher in a bioreactor compared to flasks.
Even more actually. Yeah, I think you can have 2 order of magnitude more. You can go to hundreds or even more than hundreds. Yeah, it's pretty crazy. At one point you can have some kind of cake of cells, it's really dense. Yeah, the basically a bioreactor, it's a controlled environment where you can program one and say, oh, I want the feeding to happen that way. So when you have this value of oxygen go there, then you add more feedings and so on. So it's a way to have a control settings and yeah, it's difficult to be more efficient than a bioreactor, and flask will always be lower at the end. The flask will be higher per volume and will be higher per cells, but it will be lower in total per volume because the density of the cell is so high in bioreactor. So yeah, it's a whole project by itself to optimize. So this also will take a bit of time to find the perfect condition and so on.